Hello, my name is Lorenzo Hernandez. I'm a sophomore biology major in the Riley Lab, and today we're going to be talking about a preliminary investigation that I conducted into the evolutionary history of bacteriocins produced by Enterobacter Cloacae. But before we can begin discussing bacteriocins and why it is so crucial that they're investigated further in the future, we first have to talk about antibiotic resistance. And this occurs when you have a bacterial infection, say in the human body, and you deploy an antibiotic, as has been done for the past 60, 70 years, and the antibiotic is doing its thing, it's killing off bacteria, but some of those bacteria are going to be naturally resistant to the killing effect of the antibiotics, and they have this capability due to a variety of reasons, but primarily through natural selection and evolution. And so the antibiotics kill the non-resistant bacteria and leave the resistant ones alone, basically. And the resistant bacteria now have all of this free space and have all of the resources they could ever want, and they often grow and multiply rapidly within the infection and within the body. And they even have the capability of transferring their resistance to non-drug resistant bacteria, and so the situation rapidly becomes a slippery slope of resistance. And there are currently 700,000 deaths across the world due to antimicrobial resistance, and the WHO and various other organizations estimate approximately 10 million deaths by 2050 if this situation is not resolved, or if an alternative to antibiotics is not found. And one promising alternative that we're going to be discussing here is bacteriocins. And if you've eaten cheese today, you've eaten a bacteriocin. Nisin is a common bacteriocin that has been used within various food products for the past 50 years, and it is FDA approved, of course, and it is safe for human consumption. And we're going to be discussing bacteriocins further. And nisin is just the tip of the iceberg. There are many, many more bacteriocins out there in the world for us to discover and explore. And what are bacteriocins specifically? Well, bacteriocins are antimicrobial proteins and peptides produced by some species of bacteria against closely related species. And you can think of them as a sort of defense mechanism by these bacteria, and they act as sort of the sword and shield of the bacteria, and they are often used when there's competition within the same environment, specifically or most commonly when that competition is a closely related species that will be competing for the same nutrients as the bacteriocin producing bacteria. And we see these bacteriocins produced in a variety of different bacterial species. And here we have a list of some of the most common bacteriocins currently being explored in the literature. And of these bacteriocins, the most common is colicin, which is very abundant and diverse. It is the most abundant and diverse bacteriocin that we have found so far, and a lot of research has gone into investigating these colicins. And due to this, we have learned a lot about bacteriocins, but we have also ignored some other species of bacteriocins that may reveal different things about bacteriocins in general and different things about those specific bacteriocins in terms of how can they be applied in different ways than, say, these colicins. And it is within these more obscure bacteriocins that I decided to focus my efforts on. More specifically, I focused on cloacins, which are a bacteriocin originally found within the species Enterobacter Cloacae, and it is very similar to colicins, which is what first drew my attention to it. And we can see here the crystal structures of colicin E3 compared to the cloacin CloDF13. You can see that they are very, very similar, almost identical in certain aspects. And this was very interesting to me, and I decided to focus on this more. And so, in order to further our understanding of cloacins, we must first understand and investigate their evolutionary history. Why do they look the way they do? What is the story there? And in order to do so, I first obtained as many cloacins as I can find within the literature and within public databases such as NCBI and UniPro, and I discovered 472 proteins that were recorded as quote-unquote cloacins within NCBI. And the reason I say quote unquote is because upon further investigation, I realized that many of these proteins were way too small in terms of amino acid length to be to have killing activity or to actually be a cloacin toxin. And so of these 472, only 60 of them fulfilled the necessary requirements to actually be a functional bacteriocin. And of these 60, only 6 were non-identical. The other 54 were identical to these one of these 6 proteins. And here we have a list of the 6 identified cloacins that I was able to find within these databases, and we can see that most of them are from the Enterobacter genus, and most of them are exactly 561 amino acids long. And there is only one that is 562 amino acids, and not coincidentally, when we create a phylogeny tree of these 6, it is the one outlier, and the other 5 are very closely related to each other to the point where they are so similar that even when we use different tree creating methods, this tree was created with maximum likelihood, the tree remains pretty much exactly the same. And due to this, we're not able to use this tree to infer any concrete conclusions about the evolutionary history of cloacins beyond that they are so similar to each other. And so this is a result that is very surprising because when we look at other bacteriocin types produced by other species, we can see that they're very different from each other, even when they're produced by the same species. And we're going to see this in the next slide where we compare the cloacins with colicins. And so we can see through my phylogeny tree that my cloacins are very similar to each other, but I wanted to get a quantitative analysis of this. And so to do so, I created a pairwise distance matrix between my 6 cloacins. And how you interpret this is pretty simple. The higher the number is, the higher the level of difference between the 2 proteins being compared, and the lower the number is, the higher the level of similarity. And so we can see in this value 0.00357, this is a comparison between WP1871 and WP0453. And we can see that across the board, the numbers are very low, low to a point that it was surprising to me. I knew that my cloacins were going to be similar, but not to this magnitude. And for comparison, we have a pairwise distance matrix of 6 of the most studied colicins, and we can see that usually the number is pretty high. Even when it's not to the extent that is a 2 point something, it's usually 0.3, 0.2, 0.4. It's never to the 100th place like we can see in the cloacins, except in this one outlier. But in comparison, we could see that it is pretty obvious that the cloacins are a lot similar to each other than what we see in these colicins. And all of these colicins are from the same species, so this level of difference is even more surprising. So what does this all mean for cloacins and for Enterobacter? Well, we don't really know yet. The first conclusion that we can make based on our data and on our research is that there is a mass misnaming issue within NCBI. We don't know how these quote-unquote cloacins got past the review process, but that is something that NCBI hopefully addresses in the future in terms of misnaming within their proteins, because the rabbit hole goes deeper than what we showed in this presentation. But in terms of our actual research, our main conclusion is that the level of diversity within cloacins is far from the level that we see within other bacteriocin producing genera. Using E. coli and their colicins as an example, we saw that the level of diversity there is much higher than what we see with cloacins. Furthermore, there is only one identified bacteriocin within the Enterobacter genus. This is also a surprising result when compared to other bacteriocin producing genera, where you would have E. coli that produces not only colicins but also other types of bacteriocins. And Enterobacter, from what we can see, only produces cloacin. And of these cloacins, there have only been 6 identified, and of these 6, they're almost all identical to each other. This level of diversity is staggering to consider when you think about these proteins and think about this bacteria in the wild. How did this lacking of diversity come to be, and what does it mean in terms of the ecological and evolutionary connotations for the Enterobacter genus? How does it survive out there in the wild with this low level of diversity? That is something that we have to investigate further in the future. And I thank you for listening to this project and this research. And yeah.